going a little slow. 1 minute. Alright, so welcome everyone to the Tools and Technology Seminar Series. I think everyone actually signed in. If you didn't, please do so. The sign-in sheet, I'll bring it to the back with me. But as a reminder, it's just to justify the pizza and the funded pizza and everything. Not a proprietary class or anything like that. Also, as a reminder, we do have people watching remotely, so if there are any questions are asked online, we'll try to let the speaker know about them. Also, if you ask any questions, please try to speak up to help the room microphones. That way, the remote viewers a little bit better. I think that's been helping them. So, I'd like to introduce today's speaker. We have Logan Walker, who is a PhD student in biophysics, and he's actually in Darwin Science Lab, which is located in Cell Development Biology.
And thanks for the invitation to come and tell you about some of the stuff that our lab's been working on. Like was just said, I'm a biophysics student, so not in bioinformatics, but because of the nature of the stuff we'll be telling you about today, we do a lot of bioinformatics in our lab, even though that's not necessarily the core focus. So I'm going to start out with just a couple of slides introducing kind of the problems that we work on and the kind of biological technologies that are developed in our lab, just so you get an idea of why bioinformatics is necessary for what we do to begin with. So you start off with this kind of very simple idea of what all of modern neuroscience is trying to accomplish. That's understanding that in this case, the mouse brain, but all mammalian or even non-mammalian brains, they're capable of these complex integrations and calculations where they take in all these different types of signals. In this case, I have just a couple of examples here, and they produce a bunch of different outputs, right? So like you can hear what I'm saying, you can kind of parse that information, and you can maybe later on go and download one of our tools if it helps you with your. And so how do the 71 million neurons that make up the mouse nervous system, how do they work together to do these calculations? And so to do this, we kind of need to understand what the parts list and how that parts list is put together is. So essentially, like if you've taken an introductory physics class, right, you know that this is the diagram for like a very simple electric circuit. You have a battery, a resistor, a capacitor. So when you close the switch here, it will charge up the capacitor, and then the charge on the capacitor will decay away kind of exponentially and create like a time dynamic system, right? But the only reason you're able to understand what a circuit diagram like this does is because there's just standard set of abbreviations that if you again take a physics class or this class will know what they mean. So in our lab, we're interested in how we can build a map of how neurons connect together. So in a similar sense to this electrical diagram. But rather than sending electricity, they're obviously communicating through neurotransmitters. And so in a similar way, even in this cartoon diagram as well as this very well drawn out one, you can tell that there's a bunch of different types of neurons. And so you would expect just due to the biophysics of the situation, that each one of these cells is going to respond differently. It's going to have a different set of properties. So if you want to understand how the nervous system works, you need both. You need to know the morphology and the parts list, as well as the. And so this is kind of very well summarized in this figure where. It's describing how you can categorize interneurons, the neurons that do integration. And they can be categorized basically using these four properties: morphology, connectivity, so how they're shaped, how they're connected, like I was talking about in the previous slide, as well as cell type markers and intrinsic properties. And in our lab, there's basically someone working on every aspect of this. But the stuff that I'll be telling you about today, again, and so we asked the question, can we build a better toolkit to find the wiring diagrams and morphology of neurons in So to do this, I'm going to tell you about basically 4 stories. The first one is how we use a technology called Brainbow, which you probably saw in the title, to do high contrast structural neuroimaging. This is kind of the one biological portion of it, just because if I don't introduce Brainbow, you'll have no idea what's going on for the rest of it. Then I'll be talking about a paper that was published last year about a tool called nTracer, which we use to form the quantification of the images produced in number 1. Then I'll talk about some very recent work I've been doing, creating a Python API for analysis of data being generated by nTracer, because as I'll get into, these are kind of explicitly linked. And then finally, I'll talk just for a couple of slides. I had to shorten this just to make sure that I could fit it within the hour, about adapting these technologies for high throughput imaging. Some of the kind of ongoing work that we have, basically speeding everything up for next generations. And so I'll start by saying that doing reconstructions of nervous systems have happened for literally centuries. So not quite a century ago, but for a long time we've used electron microscopy to understand neural structures, just because it gives you very fine resolution. So it gives you high resolution contrast, and you can see down here these are individual axons. It's very easy to bioinformatically separate them out because it's very high contrast. You can see in these images along each membrane you get a very dark line because of the difference in electron density along the membrane. And so you can create very high resolution maps, but as anyone who's ever tried an electron microscopy experiment can tell you, it's very expensive and it's very complicated because you have to, in this case, section your sample, so you have to actually physically slice it into a bunch of pieces, mount them on films, image each film. And so even the best labs in the world can only work on very small scale. So to put this into perspective, this figure is actually a little bit outdated because there was a paper like a month ago that beat this by a little bit in the mouse or in the fly nervous system. But in the mouse nervous system, the largest human annotated volume of nervous tissue that's ever been published is that original cube when this animation started. And you can see that at this scale, where this is the mouse brain, this is the human brain, that initial cube is so small that it doesn't even render as a single pixel. So basically, that gives you an idea that there's a scaling problem in how these problems have been addressed previously. Because again, when you're looking at this initial this Nature paper that I alluded to at the beginning of that animation, that contains 89 somas, so 89 cell bodies of neurons, and then approximately 50,000 processes, so axons and dendrites that are projecting through that volume. And that project took a team of, I think they said about 24 people, for 6 months to do. And so that gives you an idea that electron microscopy does not scale very well to the levels of creating a map of the mouse. So alternatively, light microscopy is another obvious solution. It's been used for, as I was saying, centuries. In fact, in the 1800s, when Mccroskey were doing mapping of neuron morphology, like that drawing that I showed you very early on was actually a composite of several of the drawings from some of these works. And it's very simple because even in an undergraduate biology lab, you can create slides of neural tissue, stain them, put them under a microscope, and do it in that step. But as you can see by this image here, the problem that you run into is contrast. So when you look here, you have kind of 1, 2, 3 cell bodies. And as the projections come out of the cell bodies, you can tell anywhere that they cross, there's an anticode. So anywhere that one of these branches comes up and intersects, if you are trying to come up with what the structure of this neuron is, you might accidentally go the wrong direction because there's just no way to tell those apart at the resolution that light microscopy operates at. Because obviously the diffraction limit, so how fine the physics of the resolution can go, is different between an electron beam, which is very small, relatively energy, or very high in energy, versus a optical beam. And so basically, the limiting factor for doing this kind of mapping on light microscopy is the opposite of electron microscopy, where electron microscopy is so densely, or so information dense, that you need to have teams of people working for months to produce the samples. Whereas here, it's very easy to produce the samples, but the imaging is limited by physics. And so this is really well described in this paper from late last year in the bioRxiv, where a group from the Allen Institute, which is kind of one of the leading groups doing this kind of neural mapping work, this is just one of their summary figures from the preprint. They mapped out 96 different projecting neuron morphologies, and to do that, they actually had to kill, I believe it was approximately, it was about the same number of mice, so they had to kill about 100 mice. So the number of cells that they're producing per animal is only 1. And so this gives you an idea that it's very low throughput because you have to stain the morphology so sparsely as to not have these intersection problems that I showed you in So as you can see here, like this neuron, it's very easy to tell where it's projecting to, but it has to be physically separated from this other neuron, but you don't have that. And so to deal with that, our lab uses a technology called Brainbow, where in those previous images, there's kind of like one color of labeling. In this case, it's shown as green, but it could be any color that you choose as long as there's a tag for it. And so in Brainbow, rather than using one color like green, we amplify that. So we use 3 different colors in kind of like a more traditional cassette. And that three different colors gives you 3 different possibilities for the neurons to appear as. And then when you multiply those together, because they're not mutually exclusive, right, that gives you a total of 7 different color combinations, right? So you can have a neuron that is expressing green, blue, or red, or you can have green and blue, or blue and red, and so forth. And so what this allows us to do is take images. This I just kind of like pseudo desaturated rather than actually this being the color of the image. And where this is a soma, and you can start to see that there's this problem of intersection where you can't tell where things start and end. And it turns it into something that looks like this, where even just projecting it to the human eye, you're able to tell here you have a red cell. It comes down and branches down here. And this projection, for instance, is a different color, so you know that it came from another neuron that's physically touching the soma, rather than a projection going the opposite direction. And so this is what Dahlin did during his postdoc, is kind of optimizing these technologies, and it allows us to do this neuroimaging at a much higher contrast and much higher density than traditional labeling. Additionally, I'll note that Yale Lee, a graduate student who just defended, so he's no longer a graduate student, worked on increasing this cassette capacity. So now you can have 4 different colors, or any different colors, as you will, to create this. Should not this, but you can have n different colors, which you combinatorially combine to increase this color complexity even further. And just like traditional, information theory and computer science, because these are linearly independent, you can think of it like the number of bits in a word of a computer memory. And the more bits you have, the more things you're uniquely able to identify in your image, right? So if you have 15 different colors, you can have up to 15 neurons that are physically touching without having any sort of, or without, at least in theory, having a problem of color collision. Causing ambiguity. So, additionally, as noted in the title, it didn't just say Brainbow, that expanded Brainbow tissues. Use a technology called expansion microscopy, which was originally developed at MIT. Take mouse tissue samples. This is just a diagram of a characteristic experiment where you take mouse tissue samples, you embed them in a hydrogel, So this is similar to kind of like a Jello. If you think about it in terms of consistency, the biochemistry is a little bit different. But you embed it in a gel and you hydrate it, so you increase the water or you decrease the salt concentration so that the water flows into the gel, and that gel physically expands. So you can take a piece of mouse brain tissue, physically expand it, and be able to image it at a higher resolution because now the proteins in the gel have been physically pulled apart. So by combining these technologies with Brainbow, we're able to get not only high contrast in color space, but also high contrast in physical space, as well as using this technology called MIRIX that Fred Shin, again, another prep student in the lab, has developed. We can combine it with other types of antibodies, in this case, Geoffrin and Bassoon, to mark, in this case, synapses, but you can use it for cell type marking as well. So in this case, PB and calbindin are two different types of neuron cell type markers. That kind of third column I showed you in that summary figure earlier. And so we can identify, using the combination of this and this, what the cells are expressing, how they're connecting to other cells through this kind of staining, as well as Brainbow to get your morphology information out of it. And so I'm going to show this image just because it's really impressive from Fred. And essentially, you can look at these images, and they have both a x and y dimension, but they have a z dimension as well. And so the problem that I'm going to be addressing for the rest of the talk is how do we take these huge dimensional data sets and quantify them, right? So it's really nice to look at this data, but we need to be able to get some sort of quantification out of it and reconstruct specifically the pathologies of neurons. So the challenges presented there of going from the raw data to some sort of quantification. First is that you have to coordinate the display of 4 or more color channels. So this image specifically is actually only using 3 for the Brainbow, but in those more complicated types of Brainbow that I alluded to earlier, you can have 4, 5, or 6, even more channels that you need to be able to display at once. Now it's really easy if you have 3 because you can just map it onto RGB, and it makes sense to the human eye. But once you have 4 channel, where do you map that fourth channel to? You need to be able to store these structured annotations of your images, and specifically, they form kind of branching connectivity maps. So much like computer scientists love, neuron structures can be represented as trees, but when you look at the tools that are out there for annotating biomedical images, they're not built for annotating trees. They're built for annotating lines or squares or putting points on your image. So you need to deal with that. And then finally, you need it to work inside of a kind of dynamic image processing toolkit, because even though these images look really nice when they're all done, before you can use them for science, you have to go through a pre-process and crop the images, and maybe you have to do multiple rounds of imaging, align them together. So you need this tool that you're using to be compatible with other image. So directly leading in from that, I'll tell you about our tool nTracer that does. So essentially what nTracer is a plugin for a piece of software called Fiji. If you've ever done image analysis, you've probably heard of it because it's kind of like the gold standard for open source image processing. And so nTracer is a plugin for Fiji that allows you to do this sort of structured annotation that I was telling you about a second ago. So these are just kind of keywords, but it allows you to trace, edit, visualize, and organize these annotations by code. And so because it's inside of Fiji, when you launch it up, you get a window that is literally just Fiji, as well as a couple of utility windows. Here, this is a memory monitor and a contrast control. And then these 2 windows are Fiji windows that show you your raw data and your maximum projection of your raw data. So you can use this to basically actually do your annotation here and then see a little bit better picture in the number 3. So as you can see here, there's like a process, and then if you find the same process, you can see a little bit more of it because this is adding in the frames that are above and below the Z-axis that you are looking at. And then the bulk of the program is this window right here that says nTracer at the top and is number 1. And it allows you to do all of the stuff that I just mentioned. And so I'm going to walk through just real quick some of the things it does and give. So first up here you have this bar. Because I made the window small to make it aspect ratio wise fit on the screen well, it starts to cut off the text. But basically, all of these buttons up here allow you to label the type of thing that you are tracing with nTracer. Next, you have this box right here, which stores all of the tracings that you are either in progress of doing or have completed. And because it is a tree structure, it uses basically the Java file viewing plugin, where normally this would be used to show like a file structure, but because file structures are just a type of tree, we kind of co-opt it here to give a nice interface for scrolling through these tracings. And then you down here, if you click on a certain neuron, there's all of these different labels here. These are the soma layers that have been traced. So in the cell body, it treats the cell body as a volumetric object. So these are all of the Z coordinates that you have traced, which I'll show here in a second how you actually go through. Next, you have this table, which shows you, again, the points that you've traced so that you can keep track of kind of where you are. And if you need to skip back and fix something, you can click on it here in the table, and it'll skip both your image and your editing tool back to whichever point you click on. You have a panel that allows you to actually do the editing. So all of these different buttons map to all of the operations that I've told you about before, as well as jumping around the tree. So if you want to find the last place that you haven't finished the tracing, you can use these buttons to jump to that point, or buttons like this, which will go through and try to auto-complete the soma layer that you are currently working on. There's tools to change the display features because because as you're tracing, you want to be able to see kind of where you are going with the tracing. And so by changing these overlays, it changes you can change all of the different calibers and such to make it easier to view, as well as there's some kind of like GIF and export tools that I'll show in a couple slides that these also modify. And then finally, you have the parameters for the actual algorithms underlying nTracer, which I will talk about in just a moment. And these parameters allow you to change the behavior. Finally, to deal with that problem I was saying of dealing with 3 or more channel images, you can select down here this first tab allows you to identify which image channels you want it to be doing the map on. So which image channels to actually use for the calculations, as well as in this toggle panel, choose image channels that you want to switch between, so that you can map it to a hotkey to only show a subset of your image, and then set the color that maps. So to actually use the software, you fire it up on your image. You perform your pre-processing using whatever Fiji tools that you need, because there's a plugin library of literally like thousands of different pieces of software to do different sorts of corrections and so forth. You find a location on your cell that you want to trace. You control click on it. And it does this thing. I apologize if this came through really pixelated. It does this thing called a mean shift. And so it takes a box around the place the side of that box is configurable in this panel, and it does the center of mass of the intensity within that volume, and then it shifts the square so that it tries to find the highest intensity kind of centroid. So that's what you have right here. This is the point where you theoretically clicked, and then it shifts over to here because that's where the actual neurite is, and that's where the center of mass. Same thing down here with this kind of blue. So basically, once you have your starting point, I apologize it doesn't show up very well on the projector. There's a red box right here where the white arrow's showing up. You click on another point, and it does the same process. You press S. It does. It solves the path between the starting and the end point using a maze finding algorithm. So it uses the A-star algorithm if you encountered that in a computer science class. And then you basically just repeat the process. Is that a question? And you basically just repeat the process. So you control click, press S, control click, press S, and so forth. Once you're done, you hit the complete button, which was in that user interface I showed you a moment ago, and it goes through and connects the last point that you traced to the first point you traced. And in this case, I've gone all the way around this layer. So now I have this saved in the user interface. It also supports manual tracing because since Fiji is, again, a general-purpose image processing platform, it has tools to just kind of freehand draw on your data. So we co-opt one of those to basically allow it to just either with your mouse or if you have like a drawing tablet or a touchscreen computer, just trace along the outside of the soma, and again press S, and it will convert this into a volumetric representation. And basically, as you do this, they show up in this soma panel, like I was saying. And what I did just for this example is I took this image. This is the first one that I showed you, so it shows up as Z level 59 because it's basically image slice 59 in this image stack. And I repeated it for a bunch of them. And very quickly, even just I was doing this freehand with my mouse, so that's why you see kind of like irregularities. You can start to build volumetric representations of your data. In this case, just so as I've been talking about up until now, you also want to be able to track all of the processes that are coming out of this. So again, I apologize for the appearance of the red, but in this example have clicked on the edge of the soma right here. There's a red box that appears. And clicking further down, there is a blue box that appears. You can hit a hotkey, in this case A, and it will fill in the gap between those 2 points. And you can repeat the process to trace all the way down here. Again, I apologize about the red on red because it not show very well on this projector. As you can see in this process, it kind of comes down this way and then also branches off this way to the left. So in this case, you can kind of backtrack and double click on the point right here where this blue arrow is. It will jump back in the table to where that point that you traced was and continue tracing, in this case, to the left by repeating the same process. And then as you do that, it will obviously ask you if that's what you intended to do, but it'll start to create this branch structure that that you want to build. And so in this case, there was the process that kind of came out this way, and now we have another process that's coming out here to the left that's being represented by 112, where this is the entire neuron. This is the first branch of the first neuron. This is the first branch of the first branch of the first branch, and then you can repeat this as far down as you need to go to represent some of these very complicated trees. For some of the more complicated data sets that I won't be showing you today, some of these can go out many, and it's still. So since I've joined the labs, a lot of the development of nTracer was done before I started. I've been working on adding things to nTracer to build this piece of software that we're calling nTracer 2, tentatively. The first one is I've developed some changes to the Fiji input-output system. So because these images can be very large, as you would imagine, they're represented by essentially n-dimensional arrays where you have X is one dimension, Y is another dimension, Z as the third, and then your color space as your fourth channel. These images can get very, very large, especially when they have to be basically stored roughly uncompressed. Traditionally, if you load a file into Fiji, it will do one of two things. It'll either load the entire thing into RAM and make your computer very sad if it's very large, or it'll do this other thing called a virtual stack, where as you're scrolling through the image, it will try to read individual slices of the image. Now, this is very good for your memory use. You can open a really big image on your laptop, but it is very slow because you have to basically open the file every time you change your position. So we've implemented some changes to Fiji to allow it to use kind of a first in, last out, or first in, first out stack to cache the images as it's reading through them. So if you're scrolling back and forth through the same region of the image, it doesn't actually touch your hard drive at all. It just reads it from this. Additionally, I've implemented some algorithms to speed up the visualization. So this is that same image I was showing you earlier, but after it's been traced out for every process that appears in the image. And you can see that you start to get this very complicated coloring that appears. This has been maximum projection, so it's showing like everything in the image at once rather than just showing like a certain z-plane to exaggerate that effect. But to actually draw this image, the computer has to read through the entire z-stack because it needs to know what the colors that it represents these lines as. And so by rewriting some of the internal code, there's kind of some more, in this nTracer 2.0, there's some smarter logic that goes through and subsets out the portions of the image that are important, so these high intensity regions of the tracing, and compiles them down to a hash table, which can be easily stored in memory, even for very large images, because the actual information density of one of these images is relatively low compared to the physical dimension of the image. So additionally, as I was showing you earlier, we have the ability to co-stain for synaptic markers. So there's software tools to go through and automatically identify these synapses. So these are those pre and post synapse pairs. If you've ever seen a picture of a synapse, they come in like this, and so one's marking one, the other one's marking the other. We have new informatics tools. This is just an example of the distance between these two synapses, but there's also new logging tools available for measuring color diversity and things such as that, as well as new visualization tools, which I'll show you just in a second because it ties into the. So up until now, everything that I've talked about was kind of like already published-ish. So if you have any questions, now would be like a good time before I get into the kind of like ongoing work portion.
What you described is like image segmentation or pixel level labeling problem. So is there which can be traditionally handled by convolutional neural network?
Yeah.
So is there any problem?
Yeah. So our lab actually has some preprints doing work on that with a group who was previously a postdoc and now is a professor at Texas State. So we have thought about that. But the limiting factor is that a lot of the traditional convolutional neural networks don't perform very well at like high spectral capacity. So taking off-the-shelf components and trying to do this turns out to not perform super well. We do have some like more customized approaches using cycle GANs that do perform much better, but they're still kind of in nascent states. So it's not something that you can use today. It's still in development. The other consideration is that for like electron microscopy data, there's a group at Google who's working on, they call them flood-filling networks. They're a convolutional neural network that processes through the image in a recurrent manner. So it takes a section of the image, tries to find boundaries of neurons, and then scans through the entire image doing this. But for one, those programs have only been shown to work in EM data. And for two, they're also very computationally expensive. So the only people who have actually been able to use them are Google, because they have basically unlimited capability. And even then, it takes them months to do a single. So
now, oh, sorry.
You had a
Oh, yeah. I just have a question about going back to the data collection, the expansion microscopy technique, and just the challenges of that. I mean, how hard is it to control that for like distortion and the rate of expansion if you want to compare across samples and all this stuff?
So it's been shown that the distortion is actually relatively low. Basically, the way one of the ways that that's been shown is by taking images that weren't extended and were just processed through like super resolution techniques. So if you're familiar with like Storm or STED, and comparing that directly to image or the same kinds of samples that were processed through expansion microscopy, it turns out that if you measure a lot of the dimensions of, for instance, actin filaments, they come out the same between those two methods. It turns out that at small scales, because this expansion is so isotropic, there aren't very many distortion effects as long as you control for like the actual like, physically warping the. So you just have to be careful when handling it to not like actually rip it or actually like bend it. But it's a very valid question. It basically comes down to the same concerns apply for handling these types of samples as if you were just handling like a very thin slice of neural tissue, which is something that's been routinely done for
With the changes that were made to random access of these images, they show that they're very promising, but that's after traces have already been created. But the big bottleneck is actual creation of the traces, which require a lot of access of the images.
Yeah.
So there's that like too little, too late almost.
That's a very valid point. I don't have any quantification of it, just because it's a much more, because it's like you have the human interaction. It's very hard to like quantify in a meaningful manner. But because it's using this first in first out structure, what you often find is as a human doing the data analysis, you'll scroll basically back and forth through the same region of the image a lot, because you're trying to find basically where something starts or where something ends. You'll kind of scroll through the image, identify it, scroll back, make sure that you're correct, and then actually start doing your data analysis. And so in that operation, it does end up being cached, because the first time where you're just kind of like quickly scrolling through, it sees the region of the image. It buffers that region, and then when you're going back and skipping back and forth between different Z frames, those have already been read in, because during your kind of human overview, you read through that region.
Is it a true first in, first out, or is it an LRE?
So the version that's implemented right now is first in, first out. I have some like more clever implementations that turn out it depends on how you basically have to tune them per person to actually make them faster. So it turns out that the dumb implementation ends up performing much better than something more complicated. But I've explored.
How do you interpolate continuous values over the z-axis?
So in this case, it basically just discretizes everything to the XYZ grid. And so if you notice in the tracing tables, all of the coordinates are recorded in like pixel X. So it's not interpolating. So now I'm going to be telling you about some stuff that I've been working on very recently. In particular, this kind of Python package that I've been working on called nTracer Pi, which is, as you can tell by the name, a Python extension to nTracer. Working name. If someone comes up with a more clever name. So basically, I've been telling you about how we go from raw data to this kind of tracing. Again, this is something from Fred's recent bioarchive paper, and basically, this is just plotting out all of the tracings that were done using nTracer on an XY axis. So this is a Z projection. And when you and I look at this, we can try to make some sense of what's going on here. But to actually do anything statistically and rigorously meaningful, you need to be able to turn this into a quantification that can be compare using whatever method you choose. And so in nTracer, those features aren't actually built into the software itself. And traditionally, there's basically like two pieces of software that have been used to actually do this kind of morphometrics. The biggest one, called LMeasure, was developed in was last readily updated in 2008, and it relies on a version of Java 1.6 that has again not been updated since about that time. And because of that, it can do simple analysis like counting the number of branches in the file that we export from nTracer. It's called the SWC file. You might hear me. It's just a linear representation of that tree structure. It can do simple calculations, but it is hard to use and just it's a piece of software. So there is a updated version of the same idea from the Allen Institute group that I showed you did those long-range projecting maps that the 96 neurons very early in my slides. They actually re-implemented that entire software in C to use it for their data set because they couldn't get it to work with theirs in a standardized way. But now that piece of code has the same problem where it only works well if you're using it with their set of tools, and there's very little intercompatibility. I'm going
to interject, and this is why we teach bioinformatics students.
That's a very good point. And honestly, my thinking about this was actually motivated by the way that you explained your thinking about your thesis. So it is a very, very, very good point. And so in addition, even if you go through the effort, and we actually have scripts basically set up to convert our file formats into something that these other tools will accept, which is how, for instance, this graph was produced. Even though we're able to do that fairly easily, it's just kind of a data processing task. You still have this problem where our representations, as I was showing you, we treat the cell bodies as having a volume. So we treat them as a volumetric tracing, whereas most other previous morphometric studies actually didn't acknowledge that cell bodies have volume. So they basically fit a sphere to the cell body. And so whenever you try to calculate a metric that's something along the lines of the surface area or volume of a neuron, those metrics come out completely wrong because they don't know how to parse this kind of more complicated implementation of the SWC file format. So to deal with this, I've been working on a Python library that allows you to do all of those different things in a much cleaner interface than using a command line Java tool from 2008. And so basically, I have just a couple of Python notebooks that I'll walk through that, if you're interested in API development, might spark some interest. In this case, everything's encapsulated in this thing that I have imported here as Neuron, but that's actually just an arbitrary name because I've imported it into this namespace. Essentially, you're able to import the file just by calling it very simply. It has this kind of internally this very complicated data structure that's being used by nTracer, but it's representing everything as native Python, as you would imagine. You can do very simple things like just saying .plot, and it'll call matplotlib, plot out your data, and export it. It doesn't show up very well on the black slide, but there is actually axes labels and stuff along the edge. It's just this slide is dark gray and the text is black. You can do calculations like calculating the total width of your neuron, your soma surface area. You can iterate through all of the points that you've traced using Pythonic generator syntax. And then combining all of these ideas together, you can start to do one-liners where you're counting the number of tip nodes in a certain branch of the neuron data you imported, and putting some sort of addition on it in this. So additionally, as you can imagine, you can start to do kind of population analysis, where here I took a full list of this is all of the data that was traced for Fred's recent paper. and done kind of a meta-analysis of what are the volumes of the different soma, and you can see here that it forms this. And if you have additional information like annotation of what the cell type is, you can start to do very complicated statistics using relatively little code, and particularly not leaving Python. So there's a piece of software called Blender, which you may be familiar with if you've done kind of like 3D animation or like 3D any sort of 3D image processing. That also happens to be written completely in, or the backend is written in Python. So because of this, you can import these tools and use them to do plotting, like this very complicated example in 3D into a Blender user space, and then using the Blender kind of compositing tool to make really nice animations like this. That in this case highlight that there is a depth to these tracings, and that it looks a lot better than kind of like a 2D projection does, at least in this kind of. Additionally, this is something that I've been working on this last week. You can combine it with more complicated shaders. So here, this black box is the raw data for Gay's paper that he's working on. And so we're able to show the raw data, show the tracing as it comes out of the raw data, put it into physical context. This is a fly brain. I should have said that. So if you are familiar with fly brain neurobiology, you'd be able to use that context to understand this. And then, in this case, we just lay out all of the different neurons that were traced so that we can make an argument about the cell types. So, that are very similarly shaped. These that all have these protein structures. And so because of this, because this is already in Python, the code to actually generate these animations is relatively simple. It's only 50 lines. And it's basically just importing all of the files, giving them all a name in Blender, and then exporting the 3D model to the images. And so this is something that, especially with, if any of you do image processing, there's a project called Napari that's currently pretty popular on GitHub, where people are trying to implement a lot of the features of ImageJ and some of the kind of commercial image processing tools purely in Python. So I hope that in the future we'll be able to use this as a bridge between ImageJ. So that was just kind of a preview of that that I kept short because it's definitely an unfinished story. And so finally, I'm going to be talking about adapting these technologies for high-throughput imaging, just because it's really cool, and it is the thing that we are actually working on right now. But I will say that these slides, there's only like 5 of them because I cut them because I thought that I would talk a lot less of them. So everything I've shown you today is produced through something called Confocal microscopy. Essentially, here in blue is the excitation light. So it's the laser beam that you send into your sample to light it up. And in green, it is your detection field. So in this case, it's the light that your fluorophore or your dye gives off. And everything I've shown you today is confocal. So essentially, you go through and you do a point scan of your entire sample. And at each point, you record a pixel value, and that's how you composite up one of these 2D images that I've shown you. In the last kind of like 10 years, there's a newer approach called light sheet fluorescence microscopy that has kind of taken hold as the next generation of how you do this kind of high-resolution imaging experiment, where instead of doing a point scan and scanning your focal field like this, you do a field illumination from a orthogonal axis. So in this case, you can see it comes down and excites here. And then you do your detection in parallel because now you've excited this entire plane of cells rather than just scanning it. So light sheet fluorescence microscopy is, as a result, much, much faster and while still yielding the same, approximately the same. So this is a approximation that Darwin did a while back, where if you look at like a 1 millimeter cube cortical column, that's kind of like a repeating structure in the mouse cortex. and you image it with these resolutions using a confocal microscope, it'll generate like 3 terabytes of data and take 3,200 hours of imaging, which, besides the kind of just technical difficulties of dealing with that, it's basically impossible to run a microscope for 133 days without something breaking. And so if you're going to expand this approximation to a whole mouse brain, which is which is like an order of 500 larger in volume, you would have to actually run this for 1,800 years. And because we don't want to be doing an experiment or an actual funding experiment in 18 years, we need kind of a faster way to approach the problem. So using a estimate of the speed of like a light sheet fluorescence microscope, you could do this experiment in approximately 20 minutes, and you can do this experiment in approximately 7 days. But the data sizes stay about the same. So you might have this problem where you're generating using at least our preliminary design an image that's 2,000 by 2,000, 4 of them, 100 times a second. That comes out to a grand total of 24 gigabits per second, which you're starting to get into the order of basically taking a Netflix movie, in terms of data. So if you've ever purchased a computer or a server, you'll know that in the IT world, there's this kind of computer data pyramid where at the bottom you have your tape backups, so the place where you put your archival data once you're done working on it. Or at the top you have your processor registers, your cache, and your RAM. And as you go up this pyramid, it gets exponentially more expensive, but also exponentially faster. So when you want to be able to deal with this amount of data, you have to get sufficiently high up this pyramid to deal with this. So as a result, we are currently testing a bunch of different algorithms with, again, the Tom Yen lab at Texas State, who's doing that automated machine learning approaches to tracing that I talked about a second ago. We're testing using GPUs to do real-time compression from our light sheet microscope that we are currently building. And so using a set of algorithms called D3D, which are kind of in preprint form right now, we're actually able to compress our data by about a factor of 50, which takes us from doing a Netflix movie every second to a Netflix movie every minute or two, which is a much more easily handleable amount of data. As well as we've been working with HITS and ARC to build network access lanes, so like physical infrastructure between our lab and the university network to move this data into the supercomputer as fast as possible. So I don't have any kind of slides about it because it is ongoing work, but what I'm working on right now is coming up with ways to adapt this set of tools, so nTracer, Pi, etc., to work with this sort of approach to imaging. So this sort of high throughput data analysis, and as you alluded, because these images are getting to the point where you handle it by pure communication, working. So I will close with my acknowledgments. We have everyone in the Psi lab. So specifically, these 4 generated basically, I think, all of the data that you saw today, except for the example images from the slides in the middle. So Mang, who's working on the microscope design with us, Ed Logan, who does exposure microscopy, Tom Yan, who's working on all of those different GPU-based things I told you about, Doug, who's a previous postdoc, who's now a faculty member at Ball State, who took that other image, or took the sample image that I used at a couple of things, and then our. I don't believe so. It uses a different, because the excitation physics is different, and it comes in at an orthogonal angle. the pinhole is to restrict to the z-axis, but in the case of light sheet, because you are constricting the excitation beam, you no longer need a pinhole. It's being accomplished by that orthogonality. So there's no pinhole.
Any other questions? Thank our speaker one more time.